All right, so let's wrap this chapter up with our final little view at preparing specimens for the microscope. So generally what we're going to do to prepare specimens is we're going to place a sample on a glass slide. That slide is going to sit on the stage between the condenser and the objective. How do we prepare? How do we prepare these? Well, depends on what we're going to do with them. The first thing you have to ask yourself is, are your specimens going to be living or dead? If you're looking at a pond water specimen, for instance, in a biology lab, those things tend to be living. If you're looking at bacteria in microbiology lab, those things generally, even though when you took them off the plate or out of the tube, they were living, by the time you look at them, they're going to be dead. What are the aims of the observer? In other words, what are you trying to figure out? Are you trying to see structure of the organism? Are you trying to identify the organism? Are you trying to see, can these organisms move? So all of these things need to be taken into consideration. And finally, what type of microscopy are you going to be using? In our labs on this campus, we are going to be using exclusively bright field microscopy. Dark field requires different preparations than bright field. Phase contrast is different, and fluorescence. So depending on the type of microscopy you're doing, your preparation of the specimen will vary. Living preparations for fresh mounts, you're going to place a wet mount on or in a hanging drop mount to observe as close as possible the natural state of those organisms. We generally don't use traditional hanging drop mounts. Here we simply place it onto a slide. We do have some hanging drop mounts if you want to see what one of those kind of slides look like. These where it occur where the cell is suspended in either water, broth, or a saline mixture to maintain viability and provide a medium for locomotion. In other words, with this, what is referred to as a hanging drop, protozoans and organisms are free to move. They're not being smashed together between the slide and the cover slip. A wet mount consists of a drop or two of culture placed on the slide and overlaid with a cover slip. A hanging drop is where a drop of culture is placed into a concave depression. A Vaseline adhesive or some other type of sealant and a cover slip are used to suspend the sample. And here is a picture of a typical hanging drop. These are a little bit more difficult than you would think to make, because what you have to do is you have to place the drop on this cover slip and then turn the cover slip over. I'm demonstrating with my hand. You put the drop up here on the cover slip, and then you turn the cover slip over, which allows that drop of water to hang, therefore the name hanging drop. And then you place it over this little concave area of this particular depression slide, and that gives those organisms in that water the freedom to move around and swim around as they would in their natural environment. Stains are exceedingly important, especially when you start looking at very small things like bacteria. Unstained cells in a fixed smear are very difficult to see, regardless of the magnification power and the resolving power. To see them requires that they be stained. Staining is a procedure that applies colored chemicals to the specimens. You have two basic types of stains. You have a basic stain, which has a positive charge. You have an acid stain, which has a negative charge. Bacteria have these negatively charged substances. They attract these basic stains very well. Acidic stains are repelled by most cells. One of the things that we do that we do use as a form of identification is, are they an acid-fast organism? Do they have the ability to attract and hold on to these acidic stains? Positive stain, the dye sticks to the specimen, gives it color. In the negative stain, the stain actually doesn't stick to the specimen. but settles from it a little distance from its outer boundary, forming a sort of silhouette. Negatively charged cells repel the negatively charged dye and remain unstained. The smear is not heat fixed in a negative stain, so distortion and shrinkage of the cells is reduced. These are also used to accentuate capsules, and nigrosin and India ink are used as negative stains. I don't do negative stains in the lab because both of these things are really, really icky to work with. You get them on your skin or clothes, and you stain clothes irrevocably, and you put a stain on your skin that's going to be there for several days. Simple stains only require a single dye. It is an uncomplicated process. Simple stains cause all the cells in the stain to appear more or less the same color. All you can really see with a simple stain is shape, size, and arrangement. You can't tell a whole lot more about the organism than that. A differential stain, on the other hand, uses two different stains. The first one applied is called the primary stain. The second one applied is called the counter stain. This allows you to distinguish cell types and cell parts. It is a more complex staining procedure. It requires some additional chemical reagents to produce the desired reactions. Here are some examples of simple stains. Crystal violet staining the bacterium Escherichia coli. Methylene blue stain showing the Saccharomyces yeast. These little structures here. Yeast reproduce by a process called budding, and those are cells that are actually budding. This is a new cell, new cell, new cell, new cell, new cell starting there. Here's one where the new cell is almost completed its separation. The Gram stain has been around for 140 years, was developed by Hans Christian Gram. It consists of a series of 4 sequential applications of chemicals. The first one, the primary stain, is crystal violet. This is a deep purple stain. Then we add Gram's iodine, which is referred to as a mordant. The mordant assists the primary stain. If the primary stain is trying to grab onto the cell structure and hang onto it, the mordant makes that binding more prevalent. It actually accentuates that binding. An alcohol rinse is referred to as the decolorizer. This product is used to rinse away any of this and this that's not attached to something. If it's just sort of stuck in between cells, the alcohol rinse rinses all that away. And then after the alcohol rinse, you add the counter stain. For example, in the Gram stain procedure, a stain called saffron. The Gram stain will give us different results owing to the different structures of cell walls and how it reacts to the series of reagents applied. The Gram stain is still, 140 years later, the universal basis for bacterial classification and identification. The first thing you want to know about your unknown bacteria, is it Gram positive or is it Gram negative? And these are practical aid in diagnosing infections and guiding drug treatments. The acid-fast stain differentiates acid-fast bacteria from non-acid-fast bacteria. It was originated as a method to detect Mycobacterium tuberculosis. These bacterial cells have a particularly impervious cell wall that holds fast, which fast means tightly or tenaciously, to the dye, which is called carbonyl fusion. When the dye gets washed away with acid alcohol, these acid-fast organisms hold on to the carbonyl fusion and don't let it rinse away. These are used for medically important bacteria, but this stain is also used for fungi and protozoans. This test shows us things that have a large lipid layer on their outside of their cells. Endospore stains is similar to the acid-fast stain. However, in this stain, the dye is forced by heat into resistant bodies called endospores. The stain distinguishes between endospores and vegetative, and by vegetative we mean cells that are actively growing. This is a significant way of determining or identifying gram-positive spore-forming members of the organism, like Bacillus anthracis, for instance, the disease that causes anthrax. Clostridium botulinum, the organism that causes botulism. Both of those are gram-positive spore-forming, and they are identified by using the endospore stain. Here's a gram stain showing both gram-positive and gram-negative things. The purple circles are the gram-positive organisms. The pink hot dogs are the gram-negative organisms. Acid-fast stains, again, the reddish-purple ones are the acid-fast. The blue ones, again, this slide is not focused extremely well, but all of these large reddish-purple areas, those are the acid-fast bacteria. The little blue ones, again, that don't show up really well, but I'm pointing at them with my laser, those are the non-acid-fast bacteria. Over here, the endospore stain showing endospores as being green and the vegetative cells as pink. Again, doesn't show very well, but that little dot right there, that one there, those two there, those two there, those are the green structures they are referring to. Again, doesn't show up too well. If you're looking at these PowerPoints just on the computer using PowerPoint slides, these colors may be a little bit better. There are some special stains which are used to emphasize cell parts that are not revealed by conventional staining methods. The capsular stain, which is used to observe the microbial capsule, this can be found not only in bacteria but also in some fungus organisms. It's negatively stained with India ink. So you apply the India ink, smear it across the bacteria, and the things that have a capsule, there'll be sort of a halo around the bacteria where the India ink doesn't stain, and that's where the capsule is. The flagellar stain is used to reveal the tiny slender filaments used by bacteria for locomotion. The flagella are enlarged by depositing a series of coats on the outside of the filament and then restaining it. The flagellar stain is one that takes several applications of the stain before you can start to see the flagella. And again, here are those special stains. This area here that I'm pointing to, that's the India ink that is staining. This area here, the sort of halo area around the cell, that's the capsule of this particular bacteria. The flagellar stains of Proteus vulgaris, all of these little long hair-like structures growing from the surface of all of the bacteria, those are the flagella. All right, that wraps up the lab, chapter 2. We will pick up with, next time we are together, with chapter 3, and we'll move right along, hopefully getting through chapter 5 very quickly before time for exam number 1. See y'all next time we are together. Have a great day.